God is good. God is always good. Dear students, I'm so happy to make this content for you. In this video, we are going to see about the ribonucleic acid or the RNA. So, RNA is nothing but the nucleic acid which containing the ribosugar. We have already seen about the DNA. In DNA, there will be the deoxyribosugar, but in RNA, there will be the ribosugar. It is found in large amount in the cytoplasm and a lesser amount in the nucleus. In the cytoplasm, it is mainly found in the ribosomes, and in the nucleus, it is mainly found in the nucleolus. RNA is formed of a single strand. It consists of several units called ribonucleotides. Hence, each RNA molecule is formed of several nucleotides. Each nucleotide is formed of three different molecules, namely phosphate and the nitrogenous bases, along with the ribosugar. If we see about the nitrogenous bases present in the RNA, it is quite different from the DNA. The nitrogenous bases present in the nucleic acids are purines and pyrimidines. In DNA, the purines will be adenine and guanine. It will be same in RNA also. But in pyrimidines, in RNA, the nitrogenous presence are purines and pyrimidines. In purines, there will be adenine and guanine as that of DNA. But in pyrimidines, there will be cytosine and uracil, which are quite different from DNA. In DNA, there will be cytosine and thymine, but in RNA, there will be uracil instead of thymine. The RNA molecule is a single stranded. Sometimes the strand may be folded back upon itself, and this double strand may be coiled to form a helical structure, that of DNA. In RNA, the purine and pyrimidines are not present in equal amount. There are three types of RNA. What are they means? They are mRNA, tRNA, and rRNA. mRNA is nothing but messenger RNA, and the tRNA is nothing but the transfer RNA, and rRNA is nothing but the ribosomal RNA. So now we are going to see about the messenger RNA. The messenger RNA is a ribonucleic acid which carries genetic information for protein synthesis from the DNA to the cytoplasm. The term mRNA was coined by Jacob and Menard in 1961. The mRNA forms about 3 to 5% about the total cellular RNA. The mRNA is synthesized as complementary strand upon the chromosomal DNA. The genetic message from dRNA is transcribed to this mRNA. So we already know what is transcription and translation and all. So DNA will be transcribed to mRNA. The mRNA carries the message in the form of triplet codon. Then there is a mRNA called heterogeneous nuclear RNA, which is a hybrid of mRNA, which is we can also say it as an intermediate from the mRNA. Sorry, we can also say it as an intermediate from dRNA to mRNA. It is processed in the nucleus and enters the cytoplasm through nuclear membrane. So it contains this name, heterogeneous nuclear RNA. In the cytoplasm, mRNA are deposited on some ribosomes. In the ribosomes, the mRNA act as a template for protein synthesis. The lifespan of mRNA in bacteria is about 2 minutes. In eukaryotes like human beings, it lives for few hours to the few days. You know what is the prokaryotes are nothing but the organisms which have the proformed nucleus, but the eukaryotes are the organisms which have the true nucleus. In eukaryotes, it lives for few hours to the few days. In the animal cells and the plant seeds, the mRNA is stabilized for months or years. Protein synthesis must be carried out within this lifespan in both in the eukaryotes and the prokaryotes. Based on the number of genes which an mRNA is formed and on the size of protein molecules synthesized, two types of mRNAs are known. They are monocystronic mRNA and the second one is polycystronic mRNA. The monocystronic mRNA is formed from a single cistron and it codes for a single polypeptide chain. The eukaryote mRNA are monocystronic, and a cistron is a DNA segment corresponding to one polypeptide chain. So, cistron is nothing but a functional gene. A cistron is a DNA segment corresponding to one polypeptide chain. So, it is a functional gene. And then the second one is polycystronic mRNA. A polycystronic mRNA is formed from many cistrons and encodes several different polypeptide chains. The prokaryotic mRNA are polycystronic. So in the next slide, we are going to see about the structure of mRNA. So if we take the structure of mRNA, the mRNA will have the nucleotides, polynucleotides like this. And in this, the first three codons, this each nucleotide is called as codon, and the first three codons together we called as triplet codon. uh, Based on the triplet codon only, the amino acids will be produced in the body. So here we are going to see about the structure of mRNA. So mRNA is a messenger RNA. It is a ribonucleic acid carrying information from the DNA to the cytoplasm. The mRNA is a single-stranded polynucleotide chain. So mRNA is nothing but the messenger RNA which carries the information from the DNA to the cytoplasm. Then the mRNA contains phosphoric acid, ribose sugar, purines, namely adenine and guanine, and pyrimidines, namely cytosine and uracil. The mRNA carries genetic information from DNA. The genetic information carried by the mRNA is called genetic code. The genetic code is the sequence of nitrogenous bases in mRNA. Among RNAs, mRNA is the longest one. Most of the mRNAs contain 900 to 15,000 nucleotides. As we already said, this each triplet codon is responsible for producing the amino acids. Based on this triplet codon only, the amino acids will produce in our body. Structure of mRNA. Then we are going to continue the structure of mRNA. Each mRNA contains the codons for a polypeptide chain. If the mRNA contains 900 nucleotides, the polypeptide chain synthesized by this mRNA will contain 300 amino acids, because we already discussed that three nucleotides together will form a one amino acid. Therefore, if there is 900 nucleotides, then it will together form 300 amino acids. One end of mRNA is called 5' prime, and the other one is called 3' prime. At the 5' prime, uh, there is a segment. At the 5' a cap is found in most eukaryotes and animal viruses. The cap is formed by the condensation of glycolate residue. Um, and if you saw the picture, you can easily observe. Here you can see that 5' and 3' end. Here the first thing will be cap, where this cap will protect the nucleotide from the 5' exonuclease, which is a restriction enzyme. And the third prime, you will have the poly A sequence, and this will protect the nucleotide from the 3' exonuclease. That is nothing but the restriction enzyme. And here we are seeing some non-coding regions. These non-coding regions are the regions which cannot form the amino acids. And the next one is initiation codon, which is usually the AG sequence. See, guys, based on the triplet codon only, the amino acids will produce in our body, as we already said. This AUG will produce the methionine. This AUG triplet codon will produce the methionine amino acid, which is usually called as a start codon. Here only the initiation site will occur. Here only the amino acid will start to produce, and then the other region is coding region, and then the next one is terminal codon. This terminal codon is usually will be the UAA or UAG or UGA in eukaryotes. This codons, the formation of amino acids will terminate, will stop. Okay, so these are about the structure of mRNA. Here we are saying that initiation is codon is followed by coding region, which contains the code for protein. It has average of 1500 nucleotides. The coding region is followed by the termination codon. It completes the translation. It is made up of UAA or UAG or UGA in eukaryotes. The termination codon is followed by a non-coding region. It has a nucleotide sequence of AAU, AAA. At the 3 end of mRNA, there is a polyadenylate sequence. It consists of 200 to 250 adenylate nucleotides, but as the age increases, the poly A shortens. The next point is the mRNA is synthesized from the DNA strand through the action of an enzyme called RNA polymerase. The synthesis of mRNA is called transcription, which we already discussed. Then the 16th point is mRNA carries the genetic information in the form of triplet codon from the DNA. The code decides the type of protein to be synthesized. So this also we already discussed. Then we are going to see about the transfer RNA. This transfer RNA is nothing but the RNA which transport the amino acid to the site of protein synthesis. It is the smallest polymeric form of RNA. It is called transfer RNA. It is also called as soluble RNA. The tRNA has the following salient features. We are going to see what are the features that the tRNA are having. So, number one, the tRNA is a single stranded RNA. It is smaller than mRNA. It transports amino acids to the site of protein synthesis. Each tRNA transports only one variety of amino acid. There are about 60 types of tRNA, as there are only 20 types of amino acids. Certain amino acids are carried by more than one tRNA. So it is quite interesting that as there are 60 types of tRNA, um, but there is only 20 types of amino acids in our body. So each amino acids are carried by more than one type of tRNA. The tRNA is formed of small units called nucleotides. There are about 75 to 80 nucleotides in tRNA. So this is the structure of tRNA. In tRNA also there is a phosphoric acid, sugar molecules, and the purines and the pyrimidines. As we already know, instead of thiamine in DNA, in RNA there will be a uracil. So this is a clover leaf-like structure of the tRNA. This is the clover-like structure of the tRNA. It has a four loops, and the first one is called diam, and here it is called thiamine pseudouracil CM, and this one is called anticodon region, and this is the amino acid acceptor arm. So this is the amino acid acceptor arm. This is the diam or the enzyme site, and this is the um, thiamine pseudouracil CM, or it is called as a ribozyme site, and this is called the anticodon arm or recognition site. So what is the main role of the acceptor arm? So here in the acceptor arm, and the amino acid will be acylated, and the 5' end of the tRNA is phosphorylated, and the 5' terminal residues is saligonylate. So in the D arm, it contains the dihydrouridine, which is unusual base, and in T pseudouracil CM, there will be a triplet sequence. So this symbol is called as pseudouridine, and the anticodon arm always contains the anticodon triplet in the center of the loop. Each amino acid is activated by a specific enzyme. This activating enzyme is attached by to a main loop of tRNA. Hence, this site is called enzyme site. It recognizes, catalyzes, and picks up the appropriate amino acid from the cytoplasm. That is because the main role of tRNA is to transfer the amino acid for the synthesis of proteins. This enzyme will recognize and catalyzes and pick up the appropriate amino acid from the cytoplasm for the protein synthesis. Then the activated amino acids attached to the carrier site. The carrier site is located at one terminal end. It is a 3' end. It has two free OH group of ribose sugar. It is common for RNA molecules. The next slide is transfer RNA. In the transfer RNA itself, we are seeing that the terminal loop of tRNA contains a sequence of three nitrogenous bases. It is called as anticodon. The anticodon is a complementary to the codon in the mRNA. The tRNA is temporarily attached to the mRNA at this site, and it reads the sequence of bases of mRNA. Hence, tRNA is described as a reader of the letters written in the mRNA. The tRNA is attached to the ribosome through one main loop. Hence, this region of tRNA is called ribosome site. The tRNA contains certain rare and unusual nitrogen bases, namely pseudouridine, methylguanine, methylaminopurine, inosinic acid, etc. The transfer RNA are more stable. The transfer RNA will be more stable, and this is a structure how the tRNA is formed. So this tRNA is also synthesized from the chromosomes from the dRNA. They contain usually high GC base pairing. tRNA is charged with amino acid corresponding to his anticodon and it becomes amino acyl tRNA. The amino acid is linked by an ester bond from his carboxyl group to hydroxyl group of the ribose of the last base of the tRNA. Which is this last base will be usually adenine molecule. The process of charging the tRNA is from amino acid tRNA is catalyzed by a specific enzyme, amino acid RNA synthetase. There are 20 amino acid tRNA synthetases. Ribosomal RNA is a ribonucleic acid present in the ribosomes, and it is also called as rRNA. It is insoluble RNA. It consists of 80% of the cellular RNA. It is the most stable form of RNA. The ribosome RNA is formed of a single strand. It is a polynucleotide chain. Each strand is formed of many nucleotide units. Each nucleotide is formed of 3 different molecules, namely the same phosphate, ribose sugar, and the nitrogenous bases. So here also the nitrogenous bases will be the same purine and pyrimidine. In the purines are adenine and guanine, and the pyrimidines are cytosine and uracil. In some region, the single strand is twisted upon itself to form a double helix. The helical regions are connected by inverting single stranded regions. In the helical region, most of the base complementary, they are joined by hydrogen bonds. So we already know that adenine and uracil and the guanine and the cytosine will be joined by the hydrogen bonds. So in the unfolded single strand region, base pairs are not complementary. Because of this nature, in the RNA, the purine and the pyrimidine bases have no equality. The RNA are classified into 7 types based on their sedimentation coefficient. They are the following rRNAs. So number 1 is 20S rRNA, number 2 18S rRNA, number 3 5.8S rRNA, number 4 5S rRNA. Here 23S rRNA, 16S rRNA, 55S rRNA. This all this RNA, that is 20S, 18S, 5.5S and 5S rRNAs are exist in eukaryotic cells, whereas this 23S, 16S and 55S rRNAs are present in the prokaryotic cells. In eukaryotic ribosomes, the large 60S ribosomal subunit contains 20S rRNA, 5.5S rRNA and 55S rRNA. The small 40S ribosomal subunit contains 18S rRNA. In prokaryotic ribosomes, the large 50S ribosomal subunit contains 23S rRNA and 55S rRNA. The small 30S ribosome subunit contains 60S rRNA. The rRNA is synthesized by the DNA by a process called transcription. The rRNA is transcribed on a small section of DNA called rDNA. In prokaryotes, a small section of chromosomal DNA is used for transcribing rDNA. In eukaryotes, all rRNA except 5S rRNA are synthesized on the nucleolus. The nuclear organizer contains rDNA. The rDNA transcribes 45S rRNA. It is cleaved into 8S rRNA and 28S rRNA. The 5S rRNA has an extra nuclear origin. It is transcribed from DNA located outside the nuclear organizer. Now we are going to see about the functions of RNA. RNA are the main components of protein synthesis. Many viruses has RNA as a genetic material. The rRNA present in the chromatic fiber initiates the replication of DNA. So these are the functions of RNA. So dear students, with this we are completing our RNA and its types. Thank you for